right, so I would like to welcome you to this month's nanoTalks. My name is Simon, and I will be hosting the nanoTalks this evening together with my colleague Marianne and coach Laura. Today, we will discuss the broad and also very recent topic of animal experimentation with the two experts, Marta, Roccio, and Bernhard Voelkl. So, however, before we dive into the topic of animal experimentation, I would like to briefly introduce our think tank, REACH. So I know that most of you are probably already very familiar with REACH, but for those who are joining us for the first time, REACH is a think tank of students and young researchers with the aim of linking science together with the wider society and also politics. And we do this through various formats, with the nanoTalks being one of them. And besides, we also run a blog and a podcast called Weiter Wissen, both with the aim of presenting scientific research in a slightly more understandable and free form, so that people that are not involved in academics still can access it and gain an insight into the current research. So you are also very welcome to subscribe to our newsletter and to keep up to date with the latest REACH activities. And of course, we also welcome you as a new member of REACH if you are interested. So right now we are over 300 members all around Switzerland. Now to the topic of animal experimentation of our talk today. So you as an audience actually can also become active and ask questions. So to do so, you can simply enter slido.com and then enter the code, which is nano-june22. And so I give you some seconds so you can copy the link. It's also in the description. And of course, you can also scan the QR code. You get there. So if you have any questions concerning the talk, maybe about animal experimentation in general, you can go on Slido and ask your questions. It should look something like this then. All right, now the talk was motivated in connection with the past initiative called Yes to the Ban on Animal and Human Experiments. And of course, there was a nationwide discussion about the benefits, handling, and also control of animal experiments. And now, following the vote from the 13th of February, we would like to take another look at the issue of animal experiments and also the alternatives in Switzerland. So since animal testing probably will occupy us, especially with such fundamental initiatives, I think it's crucial that the information and assumption one has about animal testing is linked as directly as possible to the research actually being conducted in Switzerland. So it's a great way to have an insight by asking the experts themselves about their research. Now, without further ado, I would like to welcome our first speaker, Dr. Marta Roccio. Marta is currently a senior scientist at the University of Zurich and will give us a presentation on the topic of human organoid models as an alternative to animal experimentation. So, Marta, with this, I would like to hand it over to you. The stage is yours.
Can you hear me now? Okay. So I will be talking mostly about how we use organoids as a model alternative to animal experimentation. But before I start with the science part, I would like to give you 3-4 slides about animal experimentation in general to put it into context. So first of all, animal experimentation is and can be controversial. And this is why it's important to have discussions like the ones we are having tonight. And it's a topic that is dear to me, and that's why I quickly agreed to give this presentation for this nanoTalks. So is or can be controversial, however, especially for the biomedical field, and everyone wants to see miracles like this one, where people that are paralyzed for many years can regain some control of their locomotion thanks to implant in their spinal cord. This is a patient in a clinical trial at the CHUV in Lausanne, for example. And this didn't happen from one day to the other, but it required electrodisplays, how to stimulate the fibers, how to improve the locomotion of the animal. And the same goes for a lot of life-saving drugs that we give for granted, but they have also been developed through animal experiments, and we should be grateful for that. So I thought I'd give you an overview of the numbers of animals that are used for animal experimentation in Switzerland, as this has been recently collected. So these numbers are collected between 2017 and 2019. And you see we have used almost 1 million mice, followed by rats, 225,000, poultry, fish, amphibians, and so forth. So if you then look at the different categories of experiments that have been performed, beside animal farming and ecology and exotoxicology, basically all the other categories are biomedical science, with the immune system, nervous system, and oncology being always at the top of the list. So the reason we have such defined numbers of animals, and we even know that 78 frogs have been used for animal experiments, is that this is a very tightly regulated aspect, especially in Switzerland. So you need to be able to do an animal experiment, you need to have a special training for it before you even apply. Then you submit a pamphlet of information to your cantonal authorities, where you will explain what is the objective of your animal experiment, if the model you have designed is suitable, what are the alternatives, if the experiment is really necessary. Then you describe very detailed what would be done, how much harm will be done. And then you have to balance the interest for the human society against the pain that will be inflicted to the animal species of interest. And then you go back and forward with the cantonal authorities until it either gets approved or rejected. So despite this tight regulation, we have a moral obligation to do better. And this is why the concept of 3R has been introduced in Switzerland and in many other European countries. And the 3Rs are the R of reduce, refine, and replace. So for every animal experiment that you plan to do, you should also try to think of how you can reduce the number of animals, still keeping the statistical power to draw conclusions, how you can modify your experimental setup so that you can reduce the stress or the pain for the animal, or you improve the monitoring. And finally, how can you answer these questions using an alternative type of experiment that doesn't require animals at all? So the Swiss 3R Competence Center was born a few years ago. They have a very nice webpage. I invite you to go have a look. There is also a lot of information also for non-scientists to get a little bit of understanding of what's going on. Now to the second part of my talk. I will use that, but in general, it's an in vitro model, human-based, that could be as an alternative to animal experimentation. So you can think of this as mini in a multiplanar organoids. So, as I said, the common feature is that they are derived by stem cells, and there are actually two types of. The first one is what we call adult stem cells. These are cells that are present in many tissues of the body, but not all, and they normally take care of maintaining the homeostasis of the tissue. So think, for example, of the intestine that is continuously replaced throughout life. We can isolate adult stem cells from the intestine and culture them as intestinal organoids in a petri dish. And this has been done not only for the intestine, but for all these organs, like you see here, the salivary gland, the lung, the mammary gland, the liver. So all this tissue contain adult stem cells for which we have the culture condition to expand them. The other category is pluripotent stem cells, and I will focus mostly here on induced pluripotent stem cells. This is a type of cell that, as the name says, is pluripotent, so it can give rise to all the tissue of the body. It's derived from somatic cells. It means that we can take a biopsy from the skin a sample of urine or a sample of blood, take out a single cell, transform it in a way that it becomes translated back in time to the equivalent stage that it had in an embryo, and it can now give rise to all the tissue of the body. So from this pluripotent stem cell, of course, we need then to guide them to differentiate back to the organ of origin. And this has been done successfully for the brain, the optic cup, for the salivary gland, and all these other organs. And I'll show you a little bit of what we have been doing about the inner ear as well. So these models are interesting, not only because they can replace animal experiment, but also because they're human specific. And we have learned from a lot of previous failure that the drug development pipeline is taking a lot of time and is very expensive. You can think about 10 to 15 years for drug development from drug discovery to being approved to the clinic, with millions of dollars going into it. And at the end, many drugs fail because of lack of efficacy and lack of safety. So they have been passing preclinical models in mouse, but then they don't function as well in humans. So if we can have a human model at the level of preclinical studies, we can already reduce the cost, identify the risk, increase the confidence that the findings obtained in mouse will work in human before we go to patients. And if you can do this in a patient-specific manner, also stratify patients for clinical trial. So I'll give you 3 quick examples of applications of organoids in general, and then focus on some of our work. So the first one is cancer organoids. So this is now being done in multiple centers throughout the world and also in Switzerland. People with solid tumors can get a resection of their tumors, as well as a biopsy of normal tissue adjacent to the tumor. This is then sent to the lab, and we can grow them as organoids in a petri dish. Because we can expand them and characterize them, we can put them in a freezer. But what we can also do of interest is sequence the DNA and identify which mutation has caused the tumor to arise in first place. And then we can perform drug screening. So in vitro, you're going to test which chemotherapy is killing the tumor cells, but not the normal cells. And then ideally give back this to the patients for a selective cancer treatment. The second example is an example of infection. I put here the SARS-CoV-2 infection, but this is the case for other viruses that have been used. So for example, at the beginning of the pandemic at the early 2020, we didn't know exactly what the virus would infect, which cell type would infect. And because we had all these organoids model already validated in different labs worldwide, people started to put the virus on their organoids. And identified which cells get. So you can use them to study the infection, but also to define new treatment that will block the infection. The third example instead is an example where you want to study a process that you know doesn't occur in mouse. So if you're studying a developmental disease that is specific to human, you can't do this experiment using a mouse model. And one of such examples is when you're starting the development of the neocortex. The human brain, as you know, is much larger than the one of the mouse. It has a lot more neurons. It's gyrified. It has all these turns, while the mouse brain is smooth and very tiny. And we know this expansion of neurons is caused by a population of cells, which are progenitor cells that will proliferate for a longer time and give rise to more neurons. So if what you're interested in is to study defects in this population, you cannot do in a mouse model ever. One of such example is here. Microcephaly is a developmental defects where the brain is smaller and it will lead to cognitive impairment. So scientists have taken IPS from these patients, generated brain organoids, and then look what happens if you have this mutation. So the bottom line is that in presence of a mutation in this gene, the neurons don't differentiate too quickly and the organoids become smaller because they lose this progenitor pool very rapidly during differentiation. So in conclusion for this first part, I think these mini organs or organoids are not only a replacement to animal experiment, but in some cases they can represent better model than animal experiment to understand the disease, to clarify mechanism of action either of drugs or of genes, and as well as to discover novel therapeutics. And then you might still need to go back to the animal experiment to look at things like pharmacokinetics, how the drug is absorbed, how you should deliver it properly, or if you're not looking at the drugs at an electrode array, like I mentioned before, where to position it and so forth. So in the last few minutes, I just give you an example of work that we are doing very quickly, more of an introduction. We work on inner ear biology. The inner ear contains the organ for hearing, the cochlea, and the organs for balance, which are the free semicircular canals and the vestibular maculae. So within the cochlea, we have a specialized cell type that you see in green here in the image. These are called hair cells, and they are the primary sound receptors. They get activated by sound, and they transmit the signal to the red cells called spiral ganglia neurons. So in this cartoon, you're gonna see how hearing works. You see the sound vibration travels through the outer ear canal, sets in motion the tympanic membrane. The ossicles of the middle ear will start to vibrate, and finally transduce this vibration to the fluid in the inner ear. And in the cochlea is where hearing occurs. So the human cochlea, for the way it's constructed, can hear frequency between 20,000 hertz and 200 hertz, depending on the location. And this is exerted by these specialized cells that I mentioned before, hair cells. They're called like this because they have hair bundles on their apical domain, and the fluid vibration induces the movement of the bundles. This activates the cells, and the cell will release a signal to activate the neurons, which then is transferred to the brain for processing. So both cell types, the hair cells and the spiral ganglia neurons, are very vulnerable to damage, and their loss results in permanent hearing deficit. This affects 5% of the world population. It's now 500 million people, 1 in 1,000 newborn, 1 in 10 adult, and then above the age of 65 is already 1 in 3. So while at birth, it's mostly because of genetic mutations or some infections during pregnancy, then the ratio of these infections, the exposure to antibiotics and chemotherapeutics, loud noise overexposure, increases throughout life, and this is why the ratio goes up so much. Plus, radicals produced during aging also cause damage to the hair cells. At the moment, the treatment option consists of hearing aids that amplify sound and cochlear implant that electrically stimulate the neurons if you don't have any residual hair cells left. But at the moment, there is no drug or gene-based therapy for hearing loss. So, but as I mentioned before, there is a lot of things in the pipeline. We have developed drug-based therapeutics, gene therapy, cell replacement strategies that have been tested in animal model, but have not yet translated to clinical trial. And this is mostly because we have no human-specific model to validate any of these findings. And ideally, you would want personalized in vitro human-specific models to do this. So my lab has been working for a couple of years now on the development of human iPS-derived inner ear organoids. These are, again, stem cell-derived 3D aggregates of multiple cell types that contain sensory epithelia, as well as the sensory neurons or otic neurons. So here you have the hair cells in yellow, the neurons in green, and another cell type called supporting cell in red. And this looks remarkably similar to what we get from a human cochlea at the equivalent stage of development. So the hope now is to use first the organoid as a bridge between the animal experiments that have already been conducted and the clinical trials to validate many of the therapies that have already been developed. And in a second step, to reduce the animal experiment that we use once we have validated this as a tool for drug development. So I think these inner ear organoids represent an advanced human-specific model to study hearing loss and ideally to validate new therapies. And I would like to thank you for your attention, and I will stay until later for questions. Thank you very much.
Thank you very much, Marta, for your talk. It was very interesting, I think. Now the second talk is held by Bernhard, working at the University of Bern, and he's giving a talk regarding the issue of validity. Yeah, thank you. Maybe you can introduce yourself and then, yeah, enjoy Bernhard's talk.
Yeah. Thank you very much, Marianne, for inviting me and, well, asking me to give a presentation here, which I'm happy to do. So my name is Bernhard Voelkl, and I'm a member of the Animal Welfare Division at the University of Bern. So this is part of the Veterinary Public Health Institute. So we're basically, yeah, doing research about animal welfare, both farm animals, but also companion animals, and also animals used in animal experiments, and ask question, what are contemporary issues, and what are the problems, how can we address them? And today I want to talk about validity of animal experiments. 2 weeks ago, I made an exciting and adventurous journey. It was exciting because it was the first travel to an international conference since over 2 years. And it was an adventure because I was traveling with Deutsche Bahn, which is always an adventure because there are delayed trains, there are canceled trains, and tight connections and missed connections. So traveling through Germany is quite an adventure. And whenever you embark on such an adventure, you get to know interesting fellow travelers who suffer from the same problems. So that happened to me 2 weeks ago. I met a woman visiting from the United States. She wanted to visit a friend in Germany, and we start talking. It was a long train ride. And when she learned that I'm a biologist, question was then, well, there were questions about the corona virus and the pandemic. And one question, RNA vaccines so fast? And my answer was, and this is basically the intro to my talk, my answer was, it wasn't developed fast. It took people over 30 years to develop it. And as I was working on my laptop anyway, I showed her this paper, and specifically this figure in this paper, to explain this. So this is a very interesting review paper by Verbeek and colleagues. It came out in September 2019, so just a few weeks before the first reports of this SARS infection in China were published. And the title of this paper is 3 Decades of Messenger RNA Vaccine Development. So it's a review, and this here is the figure that I showed my fellow traveler. And this basically summarizes the basically 30 years of development of mRNA vaccines. So according to these authors, it basically really started with a rather groundbreaking paper in 1989. So that was 30 years before 2019, by Malone and colleagues, who delivered mRNA to cells in vitro. And in this paper, they also very clearly stated that the idea is to use this mRNA in the future as a vaccine. And they basically also outlined what the critical steps are and how they could be approached. And yeah, so this was basically really the starting point for this mRNA vaccine research. And then there were many other milestones on the way. So yeah, just both 1990 is another important paper. I'll come back to that. And then, in the beginning of 2000s, there were the first in-human trials. In around 2010, there were the first real phase 1 and phase 2 clinical trials with mRNA vaccines. So it's not something that was just developed between 2019 and 2020, but there was a long period of, and actually almost always publicly funded research leading to the development of the mRNA vaccines that we have today. So yeah, and the reason I'm bringing this is because many of these several hundred studies were actually studies that used animal experiments. The first one, or very important one, was 1990. This was, for example, a study where they used mice as a model for humans. And yeah, animal experiments, that's the topic of my presentation. It's a controversial topic because animal experiments very often mean that we are harming animals. We are using animals for this research. They sometimes suffer stress, they sometimes suffer pain from this. They often, basically at the end of the experiment, they are killed. Human sentiment, humans have sentiments towards animals. We usually consider them as sentient beings, and we don't want to be cruel to them. Most of us, at least, don't want to be. And this is reflected, both in the public opinion and also in law. So here, for example, in, this, in Switzerland, it says no one shall unjustifiably cause pain, suffering, or harm to an animal, place it in fear, or otherwise disregard its dignity. And that's a rather broadly accepted notion, so which is supported by large parts of society. So don't be cruel to animals, and if you are, you should get punished. Maybe not as severe as in this Bush, Wilhelm Busch comic, but yeah, don't be cruel to animals. And in Switzerland, this is basically animal experiment. How you deal with animals is regulated by the Tierschutzgesetz, and which clearly states, okay, you should not do cruel things to animals. And a very long paper, and there are many different articles regulating what you can do and what you should not do with animals. So you should not inflict unnecessary harm or pain to animals, which you could interpret that, okay, this means you shouldn't do any experiments with animals. However, at the same time, there is another piece of legislation, that's the Swiss Constitution. And here there are some rather fundamental articles. So, and one of them, Article 20, is the article for freedom of research. So the freedom, which basically states very succinct, the freedom of scientific teaching and research is guaranteed. That's a very strong statement, and it's also interesting to see where it stands. So Article 18 is about freedom of language. You're allowed to speak any language in Switzerland without being punished, even High German. Article 19, there is a right for basic schooling. Article 20 is then the freedom of research, then there is freedom of the arts, and then there is freedom of assembly, freedom to form associations. So these are the very basic civil rights which are guaranteed, and the freedom of science is one of them. So this is a fundamental legal issue, and there is a conflict between freedom of science on the one side and basically the protection of animal welfare in this piece of legislation if you're doing animal experiments. And the question is, how can we resolve this conflict of these different legal of these different legal interests? The solution is that we have basically to find a balance. And this is done in Switzerland with a so-called harm-benefit analysis. So on the one side, researchers are asked, well, to evaluate how much harm are they actually inflicting on animals when they're doing animal experiments. And on the other side, they're asked to describe how much we will actually benefit from this research if it is done. And if the benefits outweigh the harm, that's the idea, then well, an animal experiment should be approved and is seen as acceptable by society. Now, regarding the harm, what researchers have to do is severity assessment. So there are some procedures which inflict relative mild stress or harm to the animals. There are others which are more painful. And there is, for this assessment, there are 4 different levels going from 0, very mild interventions, to 3 rather severe and painful interventions. And for almost all interventions that are around, there are already some rules and guidance where to place this with respect to how much harm you do to the animals if you do such an experiment. And researchers can basically, look this up in tables and then say, okay, this is a severity 1 experiment, this is a severity 3 experiment. And so this is the severity assessment. The other thing is researchers are obliged to observe the 3 R's Marta already mentioned. And these are replace, reduce, refine, the 3 principles, which require researchers to think of ways to replace animal experiments by alternatives that do not involve the use of animals, to, if this is not possible, to reduce the number of animals used in this experiment without compromising the quality of the research, and third, to refine the experiments in a way that, well, they are less severe for the animals and constitute, well, milder interference with the animal's welfare. So this is the principle. These are the 3 R's principle, and researchers like to basically think about them and find ways to observe. If it's not possible, then they have to explain why. So this is what is done on the harm side. When it comes to the benefits, it becomes a little bit more tricky, and this is the thing where we have recently, had many discussions. So this is what the Federal Food Safety and Veterinary Office says about the benefits of animal experiments. The benefits to society can vary greatly. New medicines, verification of non-toxicity of a substance, gain of scientific knowledge, better husbandry condition for animals, and much more. In applied research, the benefits are often obvious. However, in basic research, it is often very difficult to demonstrate a direct benefit. Nevertheless, it is particularly in this type of research that the benefits prove to be the greatest in the long term. It is primarily up to the researcher himself to convincingly show the benefits that show the benefits that his work could bring. Sorry, I stumbled across the last sentence, maybe because I noticed that seemingly only male researchers have to show this, and female researchers seem to expect, well, accepted from having to prove this. Anyway, well, as you can see, this is not a very clear guideline what researchers actually have to show. And I also cannot really agree with all what is said here. And this has the following reason. If we go back to this figure, now, this was the basically the research path for mRNA vaccine development, which is clearly applied research. There is a clear goal application for human in human medicine. So that's applied research. But for most of the time, what people are doing is basically they are using animals to understand some processes going on in the organism. And for most of the time, I would argue it is impossible to give a clear, basically answer to the question, this specific study where you're asking to use mice, what will be the outcome? What will be the benefit when you conduct this study? So for example, this study by Voelkl in 1990, in the end, it was an important stepping stone, but at the time, if Voelkl, well, do you think Voelkl and colleagues, they would have written, well, we are conducting this study because we believe that in 30 years of time, the results of our study will help to fight the global pandemic. Of course, they would never have written this. They couldn't have foreseen for what their research will be used later on. And even if they had written this, would the reviewer in the review panels would they be happy with such a statement? Surely not. So they would have said, well, that sounds a rather crazy idea. So at the time here, early on, it's you cannot really say, and this is actually a rather straightforward research pipeline here in comparison to even some other areas. But at these early stages, you can definitely not say that this individual study will directly translate into a new medicine that will help people. All the same with all these other studies or so. They are finding information, the discovery of TLRs and how this ligand works and so on. This is all interesting and necessary information, but it has itself not a direct benefit that I directly translate into medicine. Even in very late phases of preclinical research with animals, we are still far away really from the development of a drug. What we are learning is we are gaining knowledge. And this is actually the only thing which we can gain from an animal experiment. Knowledge, scientific knowledge about a specific fact. And this is basically where I slightly disagree with the Federal Food Safety and Veterinary Office. They do list gain of scientific knowledge as one benefit to society from an animal experiment, one benefit to society from an animal experiment. The only thing I would say is actually it's the only benefit from an animal experiment. An animal experiment will never directly translate into a new medicine. It always can only provide knowledge. I think this is very clear. If you look at these pipelines, even in applied research, it's always just knowledge that we gain from an animal experiment. I say just knowledge, but I think that's not a bad thing. It's just something we have to be aware of. And we have to be aware of because we have this harm-benefit analysis. And when it comes to benefits, if we ask researchers, what is the benefit? The only sensible answer that the researcher can give is we gain some knowledge from, period. Not speaking of direct application, how many human lives we will save, because at the phase where we are still doing animal experiments, we cannot really foresee how it will work out later in the human. So we are gaining knowledge. The problem with knowledge is that we cannot easily measure it. And even if we could measure it in some sort of strange unit, we could not really, weigh against harm, which can also not be measured that easily. And if it was measurable, definitely in a different unit. So this is a bit of a problem. How much worth is this knowledge? We cannot really say. But what we can do is the same what we can do on the harm side. We can ask researchers to observe this so-called 3 V's. That's the construct validity, internal validity, and external validity of an experiment. So with the 3 R's, it is basically the obligation for the researcher is to think about ways to follow these 3R principles, and if it's not possible, to replace, reduce, or refine the experiments to explain why it's not possible. And we argue that the same thing should be taken very serious when we are thinking about the benefits, which is knowledge gain, observing the 3Rs. So thinking whether the experiment as I'm doing is really the best experiment I can do to figure out what I want to figure out, if I really have the highest standards, and if the results are also representative and reproducible. So these 3 Vs, there's nothing that I've invented, but it's basically a concept that was brought up by Voelkl here in Bern. And in this publication, it's nicely summarized. And here, what is meant by construct validity, it's basically the question whether the model is valid model for the phenomenon I want to study. Is there just a superficial similarity between this disease in the mouse model and the human condition, or is it really the same type of disease? So is it a valid model? Are the measurements I'm taking good proxies for the effects I really want to study? So do I measure the correct things, the things that really matter for the disease etiology? This is all the question about construct validity. Then the internal validity, this is about cleaning my test tubes, calibrating my instruments, checking data and analysis, doing all that right. But then there is also the third thing, that's the external validity. Are my research results replicable by independent researchers? And you will maybe be surprised to hear that actually this is a big problem in biomedical research. There are many studies published in well-regarded research journals, and if people in other labs actually try to replicate these experiments, they very often fail. So it seems that these results were found once, but cannot be independently replicated. That happens. What was the target population for inference? If we're just doing the experiments with one strain of inbred mice and using only male mice, for example, is this really representative? Would other mice react in the same way? Would females react in the same way? And what does this mean if I then want to translate this into clinical research? And yeah, do the results translate beyond the target population? So these are also quite old questions about external validity. And well, basically what we demand from animal experiments is that people basically perform what I would suggest to call a harm and benefit analysis. So on the one side, I would ask researchers to evaluate the harm that they're doing with the study to the animals. So has the researcher seriously considered which harm she is inflicting on the animals? And has she put a convincing effort into reducing this harm by applying the 3R principles? That's the one analysis. And a separate analysis would be the analysis of benefits. Asking in analogy a very similar question. Has the researcher explained which knowledge can be gained from the experiment? And has she put a convincing effort into maximizing the potential gain by applying the 3Vs principle, thinking about validity of the research? You cannot really weigh one against the other because it's completely different units, or there are not really units. But what you can do in both cases is basically make an argument that you have really considered this and tried your best on the one side to minimize the harm, and on the other side to maximize the benefits in terms of knowledge gain. So this is basically the what we would call a responsible approach to animal experimentation, considering both harms and benefits and weighing in both cases separately how much do we harm the animals, but how much can we benefit in terms of knowledge gain. Okay, that's as far as I wanted to introduce this topic, and I'm looking forward for any comments or discussion. Thank you very much.
Yeah, thank you very much, Bernhard, for your talk. I think it was very, very, very interesting, giving a very good insight in the question of animal experimentation. And are there any questions, Simon?
So, pardon. Actually, we've got 2 questions. So the first, I think it tackles Marta's talk. So the question is, do organoids have the potential to replace animal experiments at all? So will we get to the point where we won't need animal experiments anymore at all?
I think you can replace a subset of animal experiments. So when it comes to studying a basic principle of development, basic principle of cell biology, basic principle of how a drug interacts with your organ, or how a virus interacts with your organ, this, I think, we would be able to implement the cultural condition to mimic the organ better and better and do this. If you want to study how an electrode array stimulates nerve fibers, I don't think we will be able to have, we can already now do what they're called assembloids. So you can fuse a brain organoid with the muscle organoid and have some nerve innervating the muscle. So you get a neuromuscular junction that will contract. But these are not yet there to mimic the real innervation of an organ. So it depends if you want to do neuroprosthetics, if you want to do organ replacement in toto, this you cannot do. You will probably have always to go through an animal experiment. But there is a number of things that I'm sure we will get better and better to do in an animal-free environment.
Mm-hmm. All right. So I think a question following this is actually, how do you validate organoid models at all? So how do you assess the quality of them?
So what we and others do is generally test the cell composition of your organoids. And this you can do by staining the organoid and checking for the different cell types that are supposed to be in that organ, checking for gene expression profiles that the cell types in that organ express. So you can take a biopsy from the intestine and your organoid, triturate them, and then compare them from a genomic point of view to see how similar or how different they are. So you try to get closer and closer to the real organ. And then, of course, you want to have function. So the more similar they are in function, the more experiment you will be able to do. The more advanced type of experiment you will be able to do. So your model is suitable for some things when it's immature, but not for the more complex experiment if the cells are not yet mature. So one of the limiting factors now is for many, many organoid models is that if you start from pluripotent cells, we get to a fetal type of organ. So not an adult organ. If you want to study neurodegeneration that occurs in patients above 70 years, your organoid is not the 70-year-old piece of brain, is a 12 weeks old piece of brain. So the responses will be different. That's, those are some of the limitations that we have. But I think that, you have to design the type of experiments with the type of model, the same way that we do with the animal, which one is the best model to address this question at this time point.
All right, so then a question, Bernhard, you mentioned, for example, the harm and benefit analysis and also the 3R principles. So do you know whether students actually get in touch with this early enough at university, or is this something a scientist has more or less to learn on his or on her own during the science conducted?
So that depends. Here at the University of Bern, for veterinary students, there is a mandatory course where they are taught these principles. And this is actually now in the first year already that they first get in touch with this. and at a later stage, they again get in touch, and all students at the PhD level, if they are going to conduct animal experiments themselves, then they are also obliged to inscribe to a course and pass a test, the LTK, it's called LTK1 module, which is the basic precondition that you're allowed to conduct animal experiments. There is no one in a Swiss lab doing animal experiments without getting a proper training regarding how to conduct them, and also the legal basis. And this also includes 3 R's principles and also questions about validity. This is also part of the regulation that people need a certain education there. And for veterinary students, definitely this starts very early on in the curriculum.
All right. So another question is, I think it addresses both of you. So are you happy with the situation in Switzerland, or is there something you would really like to improve at the moment?
Who wants to go first?
Maybe I say something that doesn't make a lot of friends, but so the issue with having countries with different rules is that in the moment one becomes very restrictive, you send out research somewhere else. So if it becomes too difficult to do experiments in your country because it's not allowed, often you have to rely on collaboration to do the same experiment you would want to do with somebody else. So if you want to check the effect of the knockout of a gene on development, you have to write the whole application. It will take you 1.5 years to get approval, and then you do your gene 1. If you want to test the gene 2 that came out while you were studying gene 1, you have to resubmit for another application for gene 2 for another 1.5 years. And then you are disadvantaged against countries that don't have such a regulation and will go twice as fast as you. So these are things that, having it more homogeneous throughout countries, it will help to balance how biomedical research is done. Not specifically for Switzerland, I mean in general.
Mm-hmm.
The question is then, how do you homogenize? Do you adjust all to the high standards or to the lower standards? What kind of standards do you want to have? Whether I'm happy with the situation in Switzerland, yes and no. I think Switzerland has a very good body of regulations, and regulations are really thought well through. And if you follow them or so, I think you can, and if you really follow them and really consider the 3 R's and so really consider the 3 V's, you can do excellent research that is really justifiable and where you can use animals for this. However, as everywhere, not all researchers always follow these follow these basically recommendations through as good as they should. This is not a specific Swiss thing. This is, I think, everywhere international. So I'm working as a journal editor for a scientific journal. And from my experience over the last year, I think I get over the last 2 years, I got about 50 research papers that I had basically to evaluate and sent into a review process. Now, out of this, that's just my personal perspective. Out of these 50 research papers, there were at least 10 where I would say there were substantial flaws in the experimental design. I've seen everything in these things, from missing control groups, badly confounded factors where you cannot really say what causes what, and underpowered studies where due to the small sample size you couldn't say much anyway. And these were really flaws in the experimental design. And I think that all this stuff, that was at the time of publication, where the authors wanted to publish their results, which means they originally got funding for the results, which means the funding proposal was already reviewed. They got approval from the institutional animal welfare officer and from the authorities wherever they did the research. I think even before they're doing the study, they maybe had even interactions with peers from their own institute. So there were several steps at which basically this experimental design should have been checked by people, whether it's really appropriate and good. But seemingly, these mechanisms are relatively often fail. Otherwise, I cannot explain why at the publication stage, we more often than it should be the case, find examples of really poorly conducted studies. Of course, a large percentage is fine. Yes, we are doing great research, but still, even 10 out of 50, for me, that's an unacceptable high level. And these are only those studies which really come to the level of publication. Don't know how many others have been abandoned halfway or so. So yes, there are good regulations and rules, but what we see when we look at the outcomes, we see that not always people really follow those.
Okay, so thank you very much. I'm looking if we have other questions. There's a question: how do we ensure that the public continues to value basic research, even when it comes at the cost of animal experiments? So I think it's about the interest of the public for such research.
Who wants to answer this?
I think events like this ones, or a science fair, or places where scientists share information with the public, are fundamental to get the public to understand what basic research brings eventually. So it's not necessarily directly linked to the animal experiment, but to understand what basic research is for, I think it's important for society to know and to get to know. And maybe even to understand better animal experiments. I think some people have in mind picture of us torturing animals. Actually, most of the animal facilities here in Switzerland are like a 5-star hotel for mice, where they are on control situation of temperature. No, but compared to the mouse that you will chase in your house, it's not. That we are keeping animals in extremely harmful condition in the first place. So I think having a clear discussion is important.
Mm-hmm.
I fully agree with this. So yeah, an open discussion and outlining basically, well, yeah, why we are doing this is definitely important, and what we are doing. I think, yeah, I agree with you, Marta, that sometimes the perception of animal experiment, people have these very cool pictures in mind that are sometimes promoted by animal rights activists. I mean, these are real pictures, such methods exist, but they're not necessarily representative. So a large deal of research is not of that highest severity scale. Actually, there are very few severity 3, grade 3 experiments in Switzerland. Most are lower severity. So that's definitely something important to emphasize in a discussion. But I think why we are doing this, I would come back, yeah, this is really a personal thing for me, because I fully subscribe to this notion that we are doing research in order to gain knowledge. Maybe some of this knowledge will be used later on for medical purposes. This is also a very valid and important motivation to do research, but there is also a lot of basic research done with a different motivation, where it's not about medical advancement, where we just want to understand life better, and that's as important as any other research. So we are creating knowledge, and knowledge is nothing else what defines our culture as humans. So yeah, I think knowledge has a very basic intrinsic value. And coming back to the question, how do we justify this to the general public? There is one comment I would like to make. Researchers are part of the general public too. And this is also a reason why I brought this one slide where I had these articles of the Swiss Constitution, because there, if you see where, at which point basically, the freedom of research is guaranteed, it's in this list of fundamental civil rights. So it's basically a right where the individual researcher, as a member of the public, is protected from the authorities. So the authorities are not allowed to forbid a person doing research. In principle, every member of the public is free to do research. So that's a basic fundamental right. Of course, when it comes to animal experiments, there is then this weighing with the harms we're doing to the animal, and of course we have to take this to consider this. But the starting point is that freedom of research is really a protected right, and every member of the public, every citizen should be very concerned about any restriction of this right. And I would start from this point of view. How can shouldn't members of the public be concerned about scientists being restricted in their freedom, as well as they should be concerned about animal welfare issues, which are, of course, also very important?
I think very important things you mentioned. And regarding the time, maybe a last question. So going back to the situation in Switzerland, maybe how is the situation international, like in other countries? And what do you think will be the situation in the future? So how will the use of animal experimentation and research will change internationally and in Switzerland? Maybe some like last statement maybe from both of you.
So I'm not an expert on this. I don't feel like giving numbers or comparing laws. I know for a certain type of experiment, it differs quite a bit on how freely you can either perform an experiment or request a number of animals. So just to give you an example for my field, people look at noise-induced deafening. So how much loud noise impacts on hearing function. And these experiments in the US can be done on awake mice. So the mouse is, all the mice are walking in the cage, and then the loud blast noise comes for 2 hours consecutively. And after the 2 hours, they are deaf. In Switzerland, this is not allowed. We have to anesthetize them. But the anesthesia interferes with the noise damage. So we are looking at 2 different models across 2 different continents. And then it becomes difficult to tell if we are looking at the effect of the anesthesia or the effect of the deafening. So, of course, I'm in favor of anesthetizing the animals, but if a large proportion of science is done a different way, because there it's fundamentally no problem to do it, then it becomes difficult to compare experiments. How to harmonize it? Of course, if you want to keep the standards high everywhere, then maybe it will cut away a lot of crap that we see in the literature of data that cannot be replicated. But it will become very, very tricky to apply homogeneously everywhere. So I don't know what you think about it, Bernhard, but.
Well, I think, better welfare makes better research, and better research means better welfare. So I think the two go hand in hand. Switzerland has rather stringent regulations, or seemingly stringent regulations, but in the end, if you really look at what the federal office asked researchers to do in their application for an animal experiment, they asked them to provide evidence that they're doing the experiment in the best way possible. So in principle, they ask and should ask for a proof that you're doing high quality research. And therefore, I'm rather optimistic that, well, in other countries, there might be different regulations, but in the end, doing better experiments, doing better research is good for animals because you're wasting less animals on inconclusive research. And yeah, if research in the future is the future driven by a demand for better quality, then I think this will automatically also mean better welfare for the animals.
So thank you very much. I think with this, we can officially come to the end of our talks today. So yeah, I would like to really thank you very much, Marta and Bernhard, for both talks. It was super interesting having an insight into research. And of course, I will follow with great interest how the situation will develop in the future. So yeah, again, thank you very much, Marta and Bernhard, for your talks.
Thank you very much for your invitation.
Bye-bye.
So, all the best. Bye.
Bye-bye.